Well, thank you for the introduction and for inviting me here. I always love to come here and to tell you about some of the things that we have that we've done at the Allen Institute and some of the new things that are starting to happen as things always continue to grow and evolve. So I'm going to be telling you about the Allen Brain Observatory, which was our first in vivo physiology open data set. And this was launched, that's very bright. This was launched about 12 years ago in 2012 when the Allen Institute for Brain Science initiated a new project called the MindScope project. Prior to this, the Allen Institute had created the Allen Mouse Brain Atlas and other types of brain atlases that were looking at gene expression throughout the brain. And what they wanted to do, what they set out to do, was to create an observatory of the mind where they would kind of focus a lot of different survey-style data sets on a single part of the brain. And the idea for these types of survey data sets is to collect and create a data set that is collected in a really standardized manner so that the data is very consistent. It creates a volume of data that is usually kind of unachievable with kind of standard experimental techniques in academic labs. And it's collected without a specific hypothesis, right? So it's designed to be a broad survey that can be used in a lot of different ways rather than to answer one single specific question. And while the previous work had been on things like gene expression, and they started doing, they had begun a connectivity atlas looking at how different brain regions were connected. The challenges with in vivo physiology is that it's so much more dynamic, right? There's so much more variability both within subjects and across subjects that you need to collect even more data than you would for an atlas in order to be able to make sense of that variability. And so they chose to focus this observatory on the visual cortex of the mouse. And to contextualize that a little bit, I want to jump back, and I assume this is somewhat familiar to many of you. So I'm going to be somewhat brief, but if there's questions, of course, raise your hand and interrupt me. But the visual cortex, has been widely studied in neuroscience because going back to the late 50s and early 60s when Hubel and Wiesel began using tungsten electrodes to record from single cells. And so this was a technology that they were developing in the 50s of having these wires that they could insert into the brain and really just nestle up close to a cell, and that they could record the currents around that cell and to record the spiking activity. And in their work, they focused on the visual system in the cat, recording either in the primary visual cortex or in the lateral geniculate nucleus, which is part of the thalamus. That's part of the visual pathway coming from the eye to the thalamus and then back to the cortex. And so, I don't know if I set up audio here. We're going to make this work. We can, if you've never seen the Hubel and Wiesel movies, they're really useful and kind of give you a good intuition of what these experiments were like. So what you're hearing are the spikes of one of these single cells, and what you're looking at is what's being projected in front of the cat on a screen very similar to this screen, probably a little bit smaller. And they move this beam of light around until they hear spikes. And they're going in like literally by hand, drawing on the screen these X's whenever the light is in a place where it drives activity from the cell, right? And they can move this light around, and there's places where the light drives the cells, places where it suppresses the cells. So they're putting X's where it drives it, and they put these triangles on these flanking regions where you see that the light is suppressing the activity from these cells. All right, so over here, they kind of realize it's a slightly wider region, and you hear that rebound when the light goes off, and so they go in. Takes them a while, but they put in the triangles, and they do this on the other side as well. Get these triangles, and then they can move this beam of light around, and what you're going to see in 1 second, they're going to reshape it a little bit. And when the light passes across this space, it's not going to fire unless it's hitting more X's than O's, right? And what that means is that when it starts to be oriented at this angle, it's going to, where it hits those X's exclusively, that's when you hear the firing, right? And so we have this orientation selective cell that only responds when the bar is oriented in a particular, at this very precise angle. And so even if it's, as we saw, even when the bar comes down from the side, up from the top, you don't hear a response, right? And so this was one of the types of receptive fields that they saw, the receptive field being the region in space and the pattern of light that is needed to drive a response from this cell. And so they used their tungsten electrodes to record from a lot of different cells. And so here's some of the examples that they saw. This is essentially what we just looked at, where you've got these X's and the triangles flanking it on either side. When they had the electrode in the thalamus, so this is earlier in the visual circuit, they had different shapes, right? They had these concentric receptive fields, again, with X's and triangles. Sometimes the X's are in the middle with the triangles on the outside. Sometimes it's inverted, where it responds to darkness in that center and bright on the outside. But you've got these concentric shaped receptive fields in the LGN. A colleague of theirs, Stephen Kufler, saw similar receptive fields in the retina. So this is in the back of the eye. His paper has a different style of figures, but there's a central region that's driven by the light and a broader region around it that's suppressed by the light and in the same kind of concentric shape. But then in the cortex, as the example that we saw, you see these elongated orientation selective receptive fields where you can kind of figure out how much the cell is going to respond based off how much light is hitting X's compared to triangles. But they also saw a different type of receptive field in the cortex where the cell still responded to these oriented edges. But in this case, you could position that edge in different locations, right? So with the example that we saw, when they put the light over the triangles, you didn't get a response. But in these cells, you would get a response kind of regardless of the position. These are 4 different positions of these bars, and you can see the spiking response of the cell is pretty strong for all 4 of these different positions. But if they orient the light at a different orientation, so it's still going through the same region of space, the same amount of light in that region of space, you don't see a response, or you see a much weaker response. So the cell's still orientation selective, but it's now phase invariant. Its position can vary within this broad region. And so one of the questions that came up from this small collection of receptive field patterns was, how do you go from these types of concentric receptive fields in the thalamus to the elongated, oriented receptive fields of the simple cells? And then from there, how do you get those complex phase invariant responses? And they postulated in their original paper that you could imagine a situation where if you had a small number of thalamic neurons, all with their concentric receptive fields that happen to be arranged in space along a particular direction, that if all of them are converging onto a single simple cell, you could have something like an AND operation that all 4 of these need to be firing in order for the simple cell to drive a response from that simple cell. And so that's how you could build from these small spots a response to an oriented edge. And they suggest another type of configuration going from simple cells to complex cells, where you could have an assortment of simple cells at different positions with different phases, and that as long as at least one of them is responding, it will drive the response of that complex cell, but it doesn't necessarily matter which of these 3 is being driven. This, of course, was just speculation, but there's been a lot of work since they suggested this. I can't do the math anymore, like 60 years ago, that has been able to show circuits from the thalamus converging just like is suggested here. And this type of feed-forward hierarchy has been really, is now really widespread in our understanding of neural networks and feature selectivity. So these examples of receptive fields that I've shown you, I showed you one little example from the retina, the concentric receptive fields in the LGN of the thalamus, and then going up to the simple and complex cells in the cortex. These are really just the tip of the iceberg of the visual system. So while this is a beautiful drawing and it is a really impressive circuit, it is in fact just the beginning of the visual system. This is what's known as the subway map, which shows that based off of connectivity of different cortical regions in cortex that are thought to be involved in visual processing. And so it starts from the retina down here. V1 is this purplish color, going all the way up to the end of the entorhinal cortex and hippocampus. And so there's a lot of different regions in cortex that are all connected to each other in these complex ways. And so one of the big questions that we've had in the field is, what's going on in all of these different parts of the visual circuit? What types of features are they responding to? How do we go from these spots and edges to object recognition, facial recognition, motion selectivity? How do all of those types of things get computed through these circuits? and how do we build these types of responses? And in order to understand that, one of the key things that we'd like is to be able to have a model that can help us predict the responses of a cell, right? It's one thing to kind of map a receptive field and to be like, ooh, this cell likes pictures of flowers in the field, but how can you actually predict that response based off of arbitrary stimuli? Can you, in order to know whether you actually understand that cell's response? And so what emerged was a model, again, going just in this early part of the circuit of the retina, the thalamus, and the primary visual cortex, that is kind of referred to as the standard model for this part. And it consists of a spatial and temporal filter, and the figure only shows the spatial filters, but there's a temporal component as well. And for the retinal ganglion cells, or LGNs, these look like our concentric spatial receptive fields that we mapped with X's and triangles when it becomes these elongated Gabor patches for simple and complex cells. But you have this, the spatial temporal feature that is convolved with the stimulus and then passed through a nonlinearity, right? And early on, you see this rectified response so that if the stimulus matches this filter, you get a response. But if you have the opposite, like if it's out of phase, the response is suppressed. And then those complex cells where we have the phase invariance, you've got this quadratic nonlinearity. All right, and so from this, we have the intuition for this is if this is one of our simple cell spatial features, if we were to show the animal a grading stimulus that aligns at that right orientation with the right width and spatial frequency, we'd expect to drive a response from that cell. But if we were to show this picture of a flower with a ladybug on it where the stem now aligns in that same location at that same orientation, we'd also expect to see a nice robust response. And so this has been kind of the standard model in the field and how we've thought about this part of the circuit for many years. And about almost, I guess, 20 years ago now, there was a book that came out called 23 Problems in Systems Neuroscience. I always like to bring up this book because if you're ever looking for project ideas, there's 23 project ideas, actually probably more project ideas, but 23 really interesting, big open questions in the field. Even though it's 20 years old, I think they're all still open problems. I should really check that though. I always say that, but maybe it's not true. Maybe it's only 21 problems. But one of the chapters in this book is called What's the Other 85% of V1 Doing? And the authors basically point out that this standard model does a very poor job of actually predicting the activity of V1, which is to say that there are cells that are well predicted for some types of stimuli. Part of the problem that they point out is that there's a lot of bias in the way we record from neurons, right? So the recording techniques, especially those single cell recordings, you're not going to put an electrode in the brain and record a really unreliable response, a really quiescent cell. You're not going to spend your time doing that. So a lot of the recordings are from cells with big spikes and high firing rates. And so there's a strong bias, particularly like layer 5 neurons. Those are big cells with high firing rates. So there's already kind of a bias for cells that already are responding the way we expect them to. There's other biases as well where we go in using gratings, right? That's what we think we're going to see. So we go in, waving these bars around, listening for these responses. And so then we're finding the cells that are responding to the stimuli that we're showing to it. There's other types of biases as well that they call out. Some of the biases have to do with the types of theories of how we think about what's going on. But one of the things that they point out is that there really is a failure to be able to go from the standard model and to predict responses to naturalistic stimuli. And so they show this example in their chapter. So this is in blue is the response of a cell to a natural movie clip that they showed to the animal. I think this is a recording from primate, but I'm not 100% sure. And then the red trace is the response that they predict based off of the measured receptive field, where they measure its orientation, its spatial frequency, all of the tuning properties of the cell. They create that spatial temporal filter, and when they convolve the movie with it, the predicted response is what's shown here in red. And so you can see this just a failure to map between the prediction and the actual response. They even show another illustration of this where the red trace here is the same as the blue trace. These are 3 cells that all have the same receptive field properties, right? So the size, the location, the orientation, the spatial frequency, temporal frequency, all of the properties of the receptive fields are really closely the same for these 3 different cells. But when you look at their responses to this natural movie clip, they have different responses. They respond, like, there's some places where they overlap a little bit, but they have a lot of differences across these 3 different cells. So this ability to predict responses to naturalistic stimuli and not just grading stimuli and things like that is a key failure of the standard model. And so in their book chapter, in their conclusion, they have this paragraph where they ask what would it actually take to understand V1, and they say there's at least 3 ingredients required. One is an unbiased sample of neurons of all types, firing rates, and layers of V1. Second is the ability to observe simultaneously the activities of hundreds of neurons in the population. And then the third is to the ability to predict, at least qualitatively model, the responses of the population under natural viewing conditions for these more naturalistic types of stimuli. And so in many ways, this little paragraph kind of exemplifies kind of the goals that we had in creating this survey of visual activity in the visual cortex, was to try and create this type of unbiased sample of neurons. Nothing can be completely unbiased, but let's say a reduced bias sampling of neurons of all different types, of all different layers in V1, and to record the activities, I hit the wrong button, of hundreds of neurons in a population, right? Not going in one by one, but trying to record as many cells at a time as we can. So the work on this pipeline and this data set, it's called the Allen Brain Observatory Visual Coding 2-photon data set. You can see our paper from a few years ago, and this work was work that I did along with Jerome and Michael, as well as a large team of technicians and scientists who helped us to build the pipeline, collect the data, process the data, analyze the data, and all of those things. And so what this survey is we wanted to be able to compare responses. We're working in the mouse visual cortex, and we wanted to be able to look at responses in the primary visual cortex as well as some of the higher visual areas. So if you think of if we go back to the subway map that I showed you, where V1 was down here, we're trying to compare V1 to some of the subway stations kind of higher up in that map. Right, so this is the surface of the mouse's cortex. You have the barrel cortex up there and auditory cortex over there. Here's our primary visual cortex, and then these are higher visual areas. And so we collected data from 6 different visual areas. And we leveraged a number of transgenic tools that are accessible in the mouse in order to collect data from specific types of neurons across different layers of cortex, as well as be able to collect data from excitatory cells as well as defined inhibitory cell types. And so we're using calcium imaging, and I'll show an example of what this looks like in a second, but what we can do in the mouse is use transgenic tools to drive the expression of our calcium indicator in particular populations of cells. And sometimes we use a broad driver, so these are what are called pan-excitatory lines, like SLC17, where it drives the expression of our indicator, our calcium indicator, in any excitatory cell. And it's expressed across all of the different layers. And so we can image at different depths in order to collect data from different layers, but there's a limit to how deep we can image with these lines because of the optics of imaging deep, right? You get a lot of scatter, and so you have low signal to noise the deeper you go. Alternatively, we can target the expression of our indicator to specific types of cells that are found in different layers or even in subtypes of neurons within the layer. So in layer 5, we have populations of cells that are cortical thalamic projecting and other cells that are cortical projecting neurons. And so these are different types of cells both in layer 5, and we can limit the expression of our indicator just to those cells, which means that we don't have the problem of the optics of expression in higher layers. So we can get good recordings from layer 5 and layer 6 neurons. And as I mentioned, we can also target these 3 major classes of inhibitory cells, parvalbumin, somatostatin, and VIP expressing interneurons. And so these have very different responses than we see in the excitatory cells. And then finally, we wanted to use a standard set of stimuli so that when we're comparing the responses of these different types of neurons in these different parts of the cortical circuit, we're comparing apples to apples, right? We're looking at responses to the same types of stimuli across these different cell types and brain regions. So our stimuli included some of these grading stimuli similar to what's been used kind of historically drifting gratings that move at different directions and speeds, static gratings that are flashed, and they have different orientations and spatial frequencies, so the width of the grating. We use a sparse noise stimulus to map the spatial receptive field. And then we've got natural images, flash natural images, as well as some natural movie clips. And then, of course, we always have some period of no contrast stimuli that allows us to look at the spontaneous activity of the cells. All right, and so this allows us to, like I said before, there's no single hypothesis here, but it allows us to ask questions about how the visual information is represented and transformed through this circuit. Are there differences across cortical areas, different cortical layers and cell types, and whether the stimulus statistics can affect the encoding properties of the neurons? All right, so this is an example of one experiment. And so we're using calcium imaging, as I mentioned. So this panel here is where you see what we're seeing through our 2-photon microscope. So we have optical access to the cortex. We are expressing a calcium indicator in a particular population of cells. And what this means is that whenever a cell fires a spike, calcium floods into the cell, and then the indicator lights up, right? It's a fluorescent indicator. And so we have a mouse that's head fixed on a wheel underneath this objective. It's an awake mouse, and you can see sometimes it runs, sometimes it stands still. All of that is just by its own free choice. There's no particular task that we're asking the mouse to perform. And so you can see different cells light up at different times, and we can start to ask questions about what types of stimuli are we showing when this particular cell or that particular cell fires up. Right? So we could do kind of the same Hubel and Weasel analysis for each cell in this field of view, and there's probably a few hundred cells in this field of view. You can't see all of them at the same time, but it's a pretty dense population. So awake mouse, you saw some of the stimuli. They're shown here. Tracking the activity, we've got a camera on the eye that gives us pupil area as well as the eye position. Mice are not foveal the way that humans and primates and cats are. So their eye movements are, they don't make as many eye movements as we do, and they don't play as big a role as they do for humans and primates, but we still want to be able to capture that. We have information about when the mouse runs, how fast it runs, things like that as well. And so I mentioned this is done in with a pipeline, so we've got teams that are doing each of these steps of creating the surgical window. We map out the different visual areas so that we can go in and target them for our calcium imaging. We get the mice comfortable being handled, being head fixed, being on a running wheel. Then we collect data. For each field of view, we return to that field of view across 3 different days because our entire stimulus set takes 3 full hours for us to show it. And so we image for 1 hour at a time. And so we come back to that same group of cells on 3 different days. And then for a given mouse, we might image from a different field of view. So the first day we might image from V1. We come back for 2 more days using all of our stimulus. And then we want to collect data from area LM, which is a different visual area, and we'll do 3 days in LM. After we're done imaging, the mouse is euthanized, and we do histology, which allows us to, A, make sure that there's no problems with the brain, that there's sometimes having the window or other parts of the surgery can have adverse effects. It's rare, but it can happen. But it also can confirm that the expression pattern for the cell line that we used was in fact what we expected to see. And because we're doing this in this standardized way, it allows us to set up quality control metrics at every stage of the process so that we can make sure that the data that we're collecting is high quality. And a lot of this that you see has to do with the animal health and the procedure at different stages of the surgery. But there's also a lot of quality controls that go into the actual session data that gets collected to make sure there's not too much motion artifacts or that all of the hardware worked as it appropriately did. So I think one of the things that often goes unrecognized is how important this type of quality control is for these type of large-scale standardized data sets. So we used this pipeline and collected over 1,300 hours of data from over 250 mice. And so you can see many fields of view here from lots of different experiments. And one of the things that you might notice is that some of these are really bright with a lot of cells in them. Others are very dim and you only see a handful of cells. And so this poses another challenge, right? One challenge is our standardized data collection. The other challenge is our standardized data processing. where we go from these raw movies, we do motion correction, we do cell identification and segmentation, we extract our fluorescent signals, we correct for neuropil, we do all of these processing steps. And we want to do this with as little manual intervention as possible because we have 1,300 hours of this data. And so one of the big things is developing that type of data processing pipeline that can handle this range of variability with some ease. And so we processed all of this data. This is a table that just kind of gives you a summary. In total, I don't know if you can see it, but there's over 63,000 neurons in the data set from 456 different experiment containers, each container being 3 sessions. And you can see how this is divided. These are all of our different transgenic lines that we used. And you can see kind of which layers they were imaged in. And so many of these target, as I said before, different populations in particular layers. These are the inhibitory lines at the bottom. And then you can see how these were surveyed across our 6 different visual areas, the primary visual cortex V1, and then these 5 higher visual areas. So not every line was imaged in every area, but we got a broad survey of several lines across all of the areas. And then in 2 of the visual areas, we got all of the different cell types within those 2 areas. So of these 63,000 neurons, I will show you one. I won't show you 63,000 because that would take a very long time. But so we can look at the types of responses that we see. So this is an example cell. This is an excitatory cell in layer 5 of V1. And we can look at its responses to the drifting gratings. So these are sinusoidal gratings that move in 8 different directions and at 5 different temporal frequencies or speeds. And we represent the response here on this plot where each arm represents a different direction of motion, and each ring is one of the temporal frequencies, the slowest one in the center, the fastest one on the outside. And then you see these red dots. Each dot is a single trial, and the color of the dot corresponds to the strength of that trial's response. So where you see this group of dark red dots over here is where the cell had a reliable response, or a strong, reliable response to that stimulus condition. Whereas over here and over there, where you don't see the dots because the response is so, there is no response, that's where the cell isn't responding, right? And so we can look at the cell and we can figure out pretty quickly that the cell is responding to horizontally oriented bars that are moving upwards. It has a weaker response to horizontally oriented bars that are moving downwards, right? There's a little response over there, and kind of these intermediate speeds. We also showed static gratings. So these are, again, sinusoidal gratings that are flashed at different orientations. And so each arm here is a different orientation. And then at different spatial frequencies. That's the period of the grating. The low spatial frequency, so the wide bars are here in the middle, and the high spatial frequency is out there. And again, these are large numbers of red dots, the dots showing the strength of the response. And so we can say that it's responding to this condition here as well as that condition there. So these are horizontal, maybe slightly angled gratings at, again, intermediate spatial frequencies, right? We can look at the responses to the sparse noise. So these are black and white spots that are flashed in different positions. And we mapped separately the responses to the white spots and the responses to the black spots. This cell in particular only had significant responses to the white spots, kind of on this edge of the screen. So you know, if this was the screen the mouse is looking at, it would be kind of like over here. We showed 119 different natural images. These were selected from a couple of different image data sets. Each image was shown 50 times, and so on this plot, each ray is a different image, and each dot is a single trial. And so these long rays are where the cell, again, responds reliably and repeatedly across many trials. And so there's about 4 different images that the cell responds to. And then we showed some different natural movie clips, and so this is like a 30-second movie clip. It gets repeated 10 times. And so this is essentially just a raster plot of the cell's activity. You've got 10 trials down here and just loops around. The outer ring is just the average of those 10 trials. And so again, you'll see there's a couple of points in here where across all 10 trials, we're seeing activity from this cell. So here's this one example. We can put metrics to this, right? We can talk about the area of the receptive field. We can talk about the direction selectivity, how strongly the cell prefers the gratings moving in one direction compared to the opposite direction. Likewise, orientation selectivity. We can put a variety of different metrics on this, and if you go and look in the paper, you'll see a lot of plots comparing these types of metrics across different areas and cell types and all of that. We can also kind of try and make sense of all of these different features that we just saw, right? So we know that the cell responded to light things kind of over on the the far edge. It responded to horizontal, slightly off-angle gratings, particularly if they're moving upwards. There were these 4 images that it responded to. These are the 4 that it had strong responses to. And you can start to do a little bit of like hand-wavy explanation about what's going on here, right? So this hill has a bit of an edge kind of right over here that might be at the same angle. Maybe the top of the tiger is also having that effect. The edge of this wing could be driving something. We can look at the movie clip when the cell is one of the times where the cell responds. The movies are all taken from the opening scene of Touch of Evil. There's this 3-minute long scene with no camera cuts, but a lot of different types of motion. I know the first 3 minutes of Touch of Evil really well, and I've never seen anything after that, so no spoilers, please. Yeah?
Can you convolve the line with the rest of the images?
Can we convolve this line?
As in look for that line
in all of these images. Well, so that's kind of by eye, right? That's what we're kind of doing. And if we look at this movie clip, right, you see that the response is right at this point where this bright, it's a street organ kind of comes into the scene, right? And so we can start to kind of hypothesize that this cell is being driven by bright edges, particularly on this side of the visual image, that you can kind of pull all these things together, right? And so the question that is essentially what I think you're asking is, can we come up with a model, for the standard model for this cell that will predict this cell's response to these different stimuli? And so there we go. So this is one of the things that we did with this data set was to implement the standard model. And the way that we did this was we used a wavelet projection, a basis set. So we've got 3D wavelets, so it's both spatial and temporal, that span kind of all of the spatial and temporal features that are within kind of the mouse's visual space. I can't tell if you have a hand up. Yeah.
Yeah. I have a question regarding the images and the videos you used. Are they controlled for spatial frequency or contrast?
So are the images controlled for spatial frequency? Is what you're asking? Yeah. So that's a great question. And they are. So we spent some time looking in the data and the literature about what kind of the range of spatial and temporal frequency tuning that we expect in the mouse based off of behavioral assays and previous studies. And so one of the things that we made sure is that the images that we were using had strong power in that range. And they all do. Yeah, so we don't want to show them images that have really, really high spatial frequency that they can't possibly see. Like, who cares? So we have a bunch of different basis functions. They cover different spatial frequencies, so you have big ones and small ones. They cover different orientations, different temporal frequencies that span kind of the whole visual space. They're followed by either linear or quadratic nonlinearities so that we can have both simple and complex responses going on. And then they all get weighted, right? And so we can train and test this data either using our natural stimuli, the images and movies, or using the artificial stimuli, the gratings and noise. And we do held out cross-validation, so we're using a part of the data to train the weights on all of these different projections and nonlinearities in order to see how well we can predict it. We're also adding in the mouse's running speed because we know that also plays a factor in the responses. And so we can ask how well does this cell that we looked at, how well can we predict its responses? And so here what I'm plotting is the correlation between the predicted response to the measured response, either for the natural stimuli or for the artificial stimuli. And we actually do pretty decently. We get a little bit above 0.4 for each of them. And if you go and look in the literature for studies that have used these types of standard models, that's pretty good for what you see out there. It's definitely not 100%, but it's pretty it's considered good performance. All right, so this is one cell out of our 63,000. What about the others? The others don't look as great, right? So there's our one cell up here. This is a density plot, so the color is telling you, how many dots are in each of these places. And really the bulk of our data is right down here, very close to 0, 0, right? We have our example cell. It's not even our best cell, right? Like I could have picked this cell out here and we'd have seen even better performance. So there's a handful of really good cells, but the vast majority of our data is not well predicted at all. So what can be going on? One of the things, and this is kind of something that jumps out really quickly, but not all cells respond to all of the visual stimuli, right? The example cell that I showed you, it had reliable responses to each of the types of stimuli that I showed you. All of the gratings, all of the like there's some feature that across trial after trial it responded to. But not all cells respond to all stimuli, and this is something that really just jumps out at you when you just look at some of the data without doing very much analysis. So I've plotted 50 cells from one experiment. This is across the entire imaging session, so it's a little over an hour. And I've just highlighted the different stimulus epochs. So these are about 10-minute blocks where we're showing different static gratings, and then for 10 minutes we show different natural scenes. Then we've got 5 minutes of spontaneous activity, and then we show more natural scenes, et cetera, et cetera. And you can very quickly see some examples of cells that are like really, really active during the static gratings that suddenly are much less active when we switch to the natural scenes. And it's not that the cell died or it fell out of plane of the imaging plane, because when we come back to the static gratings later on in the experiment, we again see really robust activity from that cell. So it seems to be somewhat selective for the static grating stimulus. You see that also here with a couple of cells where for the natural movies, this movie gets repeated 10 times. And so you have a number of cells here that have really nice periodic responses where you see these kind of 10 peaks at some feature of the movie that drives it. But then when it switches over to the natural scenes, the flash natural images, the cell's not responding with a high reliability or high response rate. And so we see this selectivity. So we wanted to kind of dive into this a little bit and look at how, if a cell responds to one stimulus, reliably to one stimulus, is it going to respond reliably to another stimulus? And our first pass was just to do a pairwise comparison, where we looked at the reliability of the cell's response to its preferred condition of one stimulus compared to the reliability of its response to another stimulus. So what that means is, consider for the natural scenes, there's one image that's going to drive the cell that's averaged across all of the trials is going to drive the cell the best, right? So you find out of those 50 trials that we showed that image, what fraction of those cells had a significant response. And that's comparing each response to a null distribution that was taken during the spontaneous activity. So let's say for one cell, it's a picture of the tiger in the water that drives it the strongest mean response. And it turns out that 52% of the trials that we showed that image, it responded to. Now we want to compare that with, for the drifting grating, there's going to be one direction and frequency of grating that drives the cell the most. Of the trials of that grating, how many did it respond to? What's the correlation between those reliabilities, right? So that's what we're looking at here. And so we've got our different stimuli, static gratings, drifting gratings, natural scenes. And we've got natural movies. We've got natural movie 1A, B, and C. And that's the same movie clip that gets repeated in each of the 3 sessions that we image from those same cells. And so the correlation across these, across like 1A with 1B and 1C, is the variability of the same cell's response to the same stimulus across different days, right? And so that gives us a bit of a baseline to think about that day-to-day variability for the same cell in the same animal to the same stimulus. And then there's 2 other natural movies that are different movie clips, but are similar, taken from the same overall movie, similar statistics and all of that. And what we see is that by and large, there's very low correlation between the reliability of a cell's response to one stimulus to its response to another stimulus, right? What that means is if a cell responds reliably to our drifting grating stimulus, I don't know if it's going to respond reliably to a natural movie or not, right? A correlation close to 0 doesn't mean that it won't respond reliably. It means I don't know. It could respond reliably, it could not. The only places where we see some correlation is between static gratings and natural scenes, and then this correlation that we talk about of the same cells to the same stimulus across multiple days. But by and large, knowing that a cell responds to one stimulus doesn't tell us whether it's going to respond to another stimulus. We took this a little bit further. This is just doing a pairwise correlation, but we wanted to ask this a little bit more broadly, and we did a clustering of this reliability across stimulus types, right? So now what we've done is we're looking at drifting gratings, static gratings, natural scenes. We're looking at the reliability for natural movies. You've already seen that there's like 5 different natural movie stimuli that get shown across all these things. We're just taking the largest reliability for any one of those natural movie clips for a given cell. And we cluster these reliabilities for each cell. and these are the clusters that we get. So there's 30 different clusters. This is just a zoom in on one of these where we see high reliability for drifting gratings and natural movies, and low reliability for static gratings and natural scenes. Now we've colored these on this heat map, and the way that we set the scale in this heat map is that we know that there's some cells that don't respond to our stimuli, right? Just going back to that raster plot that I showed, if you look closely, there's some cells that just aren't super active. So we know there's got to be one cluster of cells that aren't very responsive to these stimuli. So we find the cluster up here that has the lowest mean reliability across each of the stimuli. And we used that to set the threshold, and we took the mean of these 4 reliabilities plus 1 standard deviation, and that's how we set the midpoint of our color scale, right? And that was about 27%. So a little over a quarter of trials kind of differentiated the cells that were in this unresponsive group. And so from there, we then grouped these clusters into classes based off of the patterns that we see in terms of which of the stimuli are red, so they're more reliable, and which of the ones are blue, just based off of this threshold that we've set. And so now we can group these. And so for instance, there's about 6 different clusters here where you've got reliable responses to drifting gratings in natural movies and not to the static gratings in natural scenes, right? Now again, as we saw just a couple of slides ago, the correlation between drifting gratings and natural movies itself is 0. So you still see a lot of variability in how reliable those responses are for those two things, but both of them are just above our threshold of 27%. So now we've got these different classes. So like there's some cells that respond reliably to drifting gratings in natural movies. There's some cells that respond reliably just to natural movies. There are 4 different stimulus categories here, so there should be 16 different classes that we have. But we don't actually find cells in all 16. So this is just looking at the percentage of cells in our data set in all of the possible groups that we've done. We did this kind of with different seed conditions in order to kind of test the robustness of these results. Not all of these 16 categories have cells. Interestingly, and this was kind of a bit of a surprise to us, our largest class was our none cells. 35% of the cells in our data set don't respond reliably to these stimuli. We then see big classes here for the drifting gratings in natural movies, where we believe that the motion, the temporal correlations of the stimulus are really important, so that these cells are driven by some of these motion features. We see a big category for the natural scenes in natural movies, where we think that the spatial statistics are really important. But there's only 10% of cells in our data set that respond reliably to all 4 of these classes of stimuli that we show, right? That are these kind of all cells, like the one example that I showed you. And so if we come back to our model performance, and if we look at this and we break it down by our different classes, our none cells are all down here at 0, 0, right? If a cell's not responding reliably to any of the stimuli that we're showing, we're not going to be able to predict its responses, right? Like you can't predict unreliable responses. The cells that respond reliably to the naturalistic stimuli, they fall up here, where we're seeing not terrible predictions for the naturalistic stimuli, but not good predictions for the artificial stimuli, right? And again, they don't have a reliable response for these types of stimuli, so we're not going to be able to predict that, but we can get okay predictions on that axis. And if we look at the 10% that are in our all cell category, these, while they have pretty broad, you can see that they're well kind of matched across both natural and artificial stimuli. Their median is close to 0.4, 0.4. I told you before in the literature this would be a pretty good performance for the model. And so we can then look at these results. These are just kind of lumping all of the cells together across all of these different types and areas. This is the same plot I showed you before. I've just rotated it on its side so we can look at how this breaks down across areas. So here's V1 and then the 5 higher areas. And so in V1, for instance, it's only 20% of our cells that are these none cells. And it's RL that's almost entirely made up of the none cells, right? So this 35%, you need to take that with a grain of salt because that's really somewhat in the higher visual areas and less a thing in V1. We've got about 15% of V1 that are these all cells, like we saw. But some of the things that you'll notice, so the fraction of none cells increases. Another thing that I find really interesting is that the fraction of cells, until you get to RL, but before that, the fraction of cells that are in these blue categories, which are the natural movie drifting grading, or just natural movie, but kind of the motion sensitive cells, that remains pretty constant, even though the fraction of visually responsive cells at all is going down. So this suggests that in these areas, AM and PM, that there's a kind of a condensing of motion feature selectivity compared to some of these earlier areas. Yeah?
So, doing monkey, I'm not familiar with like different areas. Is it like a hierarchy or like
yeah?
Anatomically how they're connected?
Yeah. So this is a great question. I don't know if everybody else heard this. How are these higher visual areas connected? Is there a hierarchy, or is there something else going on? It's a bit of an open question. Some people argue that it's a hierarchy. Others argue that there's some parallel tracks going on. There's a concept of, I don't know if you've learned about dorsal and ventral streams in the visual system in primates. There is thought to be those types of streams in mouse. What I'll tell you though is that we're imaging the surface of the cortex, and so most of these areas would fall into what we think is the dorsal stream. You could argue that LM might be part of the ventral stream, but most of the ventral areas are hard to access optically. So I personally, if there's a hierarchy, it's a weak hierarchy. It's a relatively flat hierarchy. And I think there's more of a parallel processing type of thing going on. But other people in the field disagree with me. So, yeah.
So when you're looking, or this is a plot of cells that are responding like reliably to a stimulus as a whole.
Yes.
And so is there, like for example, with your dynamic movies, is there a subset of the stimulus that it does respond reliably for, but not the whole movie?
Yes, so for each of these cells, there's going to be something, if it's responding reliably, say, to the drifting gratings, there's going to be a particular frequency and direction that it responds to and not others. And that's going to be different for all these cells. And you can then go and look and say, for cells in this area, the temporal frequency tends to be higher than cells in that area or something like that. Yeah. All right, so the question is, the previous question was, how do these clusters go across layers? And so this is showing just in V1. And so you'll see we've organized it by layer and then within layers by these different cell types. And the big takeaway here is for the excitatory cells, there's not a ton of difference in the functional clusters across the different layers, with a few exceptions, but generally they look pretty much the same. The big differences are with the inhibitory types. And so mixed in here, you'll see this is the somatostatin inhibitory cells. And one thing you'll notice with somatostatin is that there's almost no none cells. Somatostatin cells have really, really reliable responses, and almost half of them are the all cells, that they respond to all of the different stimuli that we show. The VIP cells, they're also interesting in that they have kind of this typical 20% are none cells, but they have a really big fraction of cells. This is another VIP in superficial layers, a really big fraction that respond to the natural scenes, natural movies. So they're driven really well by the naturalistic stimuli. They're actually suppressed by the gratings that we show, these high contrast gratings that will actually kind of often suppress them below their baseline firing rate. So you don't see much in terms of the dark blue that has the drifting gratings in there, because they don't respond to those. So those are the biggest differences we see across cell types. And likewise across layer, there's not much, there aren't really big differences that jump out among these features. All right, so a couple of conclusions that we take from this. As we pointed out, only about 10% of these neurons are these kind of textbook all cells where you see a pretty nice match between their responses to artificial and natural stimuli, where we can pretty well predict what they're going to do using the standard model. But instead, there's a lot of neurons that aren't responding to these stimuli. And this raises a lot of questions, right? In terms of what are they doing? Are they responding to more complex visual features? Or is it that they are involved in, say, the integration of different types of modalities? And things that I can say that speak to this. I think, if any of you have paid attention to mouse physiology in recent years, one of the things we see a lot of is the influence of motor activity on neural activity, even in these sensory areas. So if the mouse is running, it increases the activity of many cells and increases their visual responses as well. So for instance, we could ask, and one of the things that we looked at is, whether cells that are none cells are more correlated with the mouse's running behavior and thus less involved in the visual features. And so you can see here, this is a plot of the correlation of cells' activities with the mouse's running speed, so just ignoring visual stimuli. This is just in V1 broken down by the different cell types in the different layers. But when we look at this across our different cell classes, we actually see that the none cells, their correlation, the median is really close to 0. Our all cells actually have a somewhat stronger correlation with running than the none cells do, in fact. So it's not that they're driven by the running behavior. We could also look at the eye movements. I mentioned that mice make eye movements. Their eye movements are less frequent. They basically move just like back and forth. You can see that in this plot. Like there's very little vertical motion. It's just kind of along the x-axis. And we did a study looking at cells' activities relative to these saccades, right? And you can see examples here of cells that have increases of activity with saccades, other cells that are suppressed, their activity is suppressed across saccades, and then also cells that have like a direction selectivity, like they respond to saccades in the temporal direction, but they don't respond to saccades in the nasal direction. So here's an example of one, and here's an example of the ones that respond in the opposite direction. And so we asked again whether these saccade responsive cells are more likely to be among our none category or the other ones. And we actually saw there's no difference in our distribution in the fraction of saccade responsive cells, as well as the distribution across these different types of responses across the different categories of cells that we have. So it's a little bit above 10% of all cells are saccade responsive, and that's pretty even across our different categories. The slight differences you see here really just are sample size differences that come from that. So we don't have any reason to think that these none cells are motor driven and aren't part of in a way that makes them kind of a separate type of thing. But I think there's a lot more space to be explored about all of this fraction of cells that aren't engaged with the types of visual stimuli that we've used. And then another question, so pointing out these things, but one of the big questions is, can we develop better models to capture the neural responses that might be able to account for some of the things that the standard model is failing to capture? And I feel like this is, if you're looking for me to tell you, yes, here it is, you're going to be sorely disappointed, because this is, I feel like, the challenge in our field right now. And it kind of comes back to, as I mentioned, the 85%, what is the other 85% of V1 doing? That there are these biases in a lot of our theories, as well as the biases in the types of stimuli and the types of recording methods, that it's possible that we're approaching this kind of in the wrong angle, and that the types of models that we're using are kind of stuck in an archaic way of thinking, and we need to kind of break out of those models. And this kind of, I think, leads us into kind of where our some of the challenges in our field, and I think a lot of why I'll get you in a second. Why I think what I want you guys to pick up, right, and run along with is really trying to push new directions in our understanding and our thinking of these neural activities. And I want to point you to this review article from a couple of years ago that the title being "Large-Scale Neural Recordings Call for New Insights to Link Brain and Behavior." And it's a great review article that points out that a lot of our efforts in recent years, where we've increased the numbers of cells that we record from, this is what's called a Stevenson coording plot, showing the number of simultaneously recorded neurons as a function of year. And it was, for a while this was doubling every 7 years. I think that's now, I think we've broken that with new technologies that have been developed through the Brain Initiative for dense electrophysiology and for optical imaging methods, where we're really starting to deviate from that line. But we're able to collect these really large-scale data sets, and when we apply a lot of the analytical methods that were developed when we were recording 1 cell at a time or 4 cells at a time, a lot of those ideas are starting to fall apart. And so the challenge in the field is really to kind of push further and to use new analytical methods to approach these data kind of from new vantage points. And so I kind of put this to you as I'm looking to you to help answer these questions, because the methods I learned back in back over here when we were looking at a dozen cells at a time, and I can't do the math for thousands of cells at a time. That's just outside. I'm too old to learn new tricks. So I'll give you the data, and you guys come with new analyses. But I think that this is then kind of, actually, before I shift, you had a question a second ago.
On the non-cells,
Yeah.
are the neurons in the higher visual areas less responsive in general?
So the neurons in the higher visual areas. So the final visual area I showed you, RL, that had 85% of non-cells, I would put like a big grain of salt next to that area. It's a really kind of weird area, and it's hard to kind of map it on the surface of the cortex. So it's possible we didn't target it very well. And so I'm always like, when people are kind of digging into the higher visual areas, I'm often like, maybe ignore RL in our calcium data set. There's some like peculiar challenges with that particular area. The other areas, I think I can jump back real quick. These other areas, like there's still 60% of cells that are responding to something among these stimuli. And I think there's again big questions about what are these other 40% of the cells doing. And like I said, I think that's a big open question. Yeah.
First, thanks for the very nice set. I mean, it's amazing to see so much data about physiology of non-visual cortex. I have a question that regards like the shifting paradigm that it seems that this data proposed.
Yeah.
Like that we began from Uber and Wiesel 50 years ago from a tuning based paradigm where we search for these nice tuning curves of these neurons describing what is stimuli, but now this is not working anymore. Some people say that populations are the way forward, but the population seems like a sort of weird term. What do you think about this?
Sorry, that population recordings, did you say?
Analysis.
The population analysis to understand the
Instead of analyzing a single neuron, you analyze a population of neurons.
Yeah.
Behavior.
Yeah, no, I mean, I think that that's a big thing, and this is where I think that the new technologies, I think, are really kind of pushing us into a new space, right? Because we now can have really large populations that aren't a dozen cells, but are hundreds and sometimes thousands of cells, often from different brain regions simultaneously. And I think that that's really going to open a lot of new avenues for analysis and new kind of questions that we can really drive our understanding further forward. So I absolutely think that the population level is the place to look. I do think at some point you need to kind of bring that back to single cells a little bit and understand how single cells are part of that. But this is a question, and it kind of goes to like a slightly more philosophical space of like, at what level do we need to understand how the brain works, right? And is it at the cell? Is it at the molecule, right? Like, do we need to get down to the atoms? And so, personally, I like to be in that cell to circuit range. That's kind of where I'm most comfortable. But I feel like maybe we need to shift and be looking at the circuits and the populations, and then, in 15 years, come back and understand how the cell is part of those circuits a little bit better. Or we have people answering different parts of those questions simultaneously. My comfort space is I want to understand how a cell is part of a circuit, but I don't know that that's the only place we can answer any of these questions at. Yeah.
I work on human data, so I'm not exactly sure, like, if I guess functional connectivity is as much
It's a huge thing.
And so I guess my question is, with these, I guess, selective responses within each of these areas, do those are these like selectivities then functionally connected across the layers of the hierarchy, or is it that like these cells it's just layer selective?
Sorry, rephrase that just slightly for me.
So is it that like if cells in V1 or that are selective to the static gratings are more preferentially connected to the gratings in LM?
No, great. That's a great question. And this is the thing where this data set isn't really well poised to answer that question because we image each area separately. And I can jump real quickly. The big challenge is, okay, so I'll come back to your question in 1 second. So this is one data set that we collected, but there's a couple of related data sets to it. And so one is a data set that uses essentially the exact same stimulus, some slight modifications, but now we're using neuropixel electrodes. So these are silicone probes that have hundreds of sites along this shank. And so you can record thousands of neurons along a single neuropixels probes. And then we used 6 neuropixels probes. So we're hitting each of those visual areas that we did our calcium imaging in simultaneously. And then we're getting the cortical areas as well as subcortical areas simultaneously. And so this is the type of data set that might allow you to ask that question a lot better, where you can look for cells that are selective for certain types of features or certain types of properties, and then look for correlations of those cells with cells in different areas. Whereas in ours, we're just one layer, one area at a time. And so, yeah, so this is, and this kind of points out why kind of having these different modalities can be really valuable. Like here, you don't get the cell type resolution that we had for the calcium, but you get this simultaneous area. You get spike time resolution, which you don't get with calcium imaging. We have 2 other data sets. Oh, here's just a snippet of some of the things that they saw with this data set is asking the question about the hierarchy, right? And so here they analyzed these data, looking at certain features. This is the latency of the first spike, and showing kind of a progression of that latency as you kind of move across these different visual areas. I don't know if you can see down there, but suggesting that there is kind of a linear hierarchy among these areas. We have 2 other data sets that some of my colleagues collected, again, leveraging the same pipeline, so the same infrastructure, the same surgeries, the same recording hardware. And here, rather than showing these passive visual stimuli, the mice were engaged in a visual change detection task. So they flash a sequence of images, and then it's the same image that gets flashed repeatedly, and then it switches to a different image. And the mouse's job is to report every time that there's a change from one image to the next. And so these experiments were done again, either using calcium imaging or using the neuropixels recordings. And one of the things that they see is they train the animals with a particular set of images. There's like 8 images that they switch between. And then when they're actually doing the recordings, they'll record once with the images that they've been trained with, and then they'll switch to a novel set of images that they've never seen before. And one of the things that they've noticed is that the response to a changed image is much larger in excitatory cells when those images are novel than when they're familiar. And they're working out some interesting things in the inhibitory microcircuit that they think kind of cause this change, this novelty response that comes out of it. You have a question?
Yeah. What about anesthetized animals?
Yeah.
Okay. But how do these statistics relate also to that kind of condition?
Yeah, so all of the data sets that I've shown you have been done in awake animals. And partly because we want to understand the awake circuit, right? And there are some noted differences with anesthetized. And we did have a study very early on when we were kind of getting all of these things built together, where they did extracellular electrophysiology, not neuropixels per se, but just extracellular probes in mice when they were awake and when they were anesthetized. I think sometimes in the same mouse, but I'm not 100% sure on that. And there are some subtle differences, mostly like in the temporal frequency tuning, but the differences are kind of subtle. You kind of have to dig into it. But there is effects of arousal on responses, both in terms of overall activity, in terms of like the strength of the responses to visual stimuli. And this is where the running comes into play, because mice are more visually responsive and more alert when they're running than when they're standing still. It's kind of a state of stupor a little bit often when they're stationary. And so there are effects of that arousal on a lot of these processes. And that's one of the reasons that we do this with the running and taking all of those things into account. Yeah.
For the novel stimuli, is it that more cells are being activated at that time, or is it the same rough number of cells that are being activated in the familiar case, but at a larger?
So there's 2 things, if I recall correctly, there's 2 things going on. And one is that, and I think this is an example of a cell that responds to familiar images and responds more to novel images. But there's also cells that will respond to the familiar images and not to the novel, and vice versa. And so there's kind of a combination of things going on. But the effects with the inhibitory circuit, with the novel, cells that respond only to the novel, it could just be the images that are chosen, right? Whereas with the inhibitory cells, they respond to pretty much all of the images, but they respond a lot differently when it's familiar versus novel. And so that's where we see that there's something going on that's affecting the gain in that circuit because of the novelty situation.
So when you show the stimulus classes, did you show them like blockwise manner? And also what's your interstimulus interval?
I just can't hear you.
Oh, sorry. So when you show the different stimulus classes,
Yeah.
did you show them blockwise or
Yeah.
completely random? Oh.
Yeah, block. So it'd be like 10 minutes of images, 10 minutes of gratings, 5 minutes of movies. They were in pretty large blocks.
Interstimulus interval.
Say that again?
What's the interstimulus interval?
So it varies. So like for the drifting gratings, you have a grating that, each grating condition is shown for 2 seconds, and then there's 1 second of mean luminance in between them. But with like the flashed images, there's actually no inter-trial interval. So it just goes from one image to the next, which is different from the behavior configuration, but in the way that we did it. And same with the static gratings, it just went from one grating to another. Yeah.
When you do your recording and you're measuring reliability, is like all the recording done on the same day?
So there's 3 days of imaging. And the one stimulus that's repeated across the 3 days is one of the natural movies.
Gotcha.
Yeah.
I guess my question then is like, if you like analyze it within day but versus across day, like, do you see a higher amount of like selectivity within a day, and then does that change across the days?
So if I jump back, if I jump back to this, right? So the real place where you can ask that question again is with this natural movie, where you can see kind of that day-to-day variability. The things that I'll point out, for instance, is that like the static gratings and the natural scenes, those are on the same day, right? And so I do think that that's why there's a slightly higher correlation. But like the drifting gratings are on the same day as natural movie 1A and natural movie 3. And there we don't see that because they're on the same day, they all respond together type of thing. So I don't think that's a huge component, but I think it's something that warrants more attention. All right, I wanted just to tell you real quickly, because we're pretty much out of time, but I wanted to tell you just a little bit about like all of this is work that has been done. These data sets are available to access. They're on the cloud. You can download them. We've got a software kit that helps you find and work with the data. But I want to tell you a little bit about kind of where things are going now and what's happening. And this was, as I mentioned at the start, this was a project that started in the Allen Institute for Brain Science. And when it started, the Allen Institute for InScience was the institute. And since then, there's now, I can't count, 7 different institutes within the Allen Institute. So there's the Allen Institute, which has the Allen Institute for Brain Science and the Allen Institute for Cell Science, the Allen Institute for Immunology. And just 2 years ago, we started the Allen Institute for Neural Dynamics. So this is a new brain science institute within the umbrella. We collaborate and work very closely with our colleagues in brain science. Brain science is really focused mostly right now on kind of questions of transcriptomics. They've done a lot of spatial transcriptomics and kind of identifying real fine resolution cell typing across the entire structure of the brain. So you no longer focus just on the cortex. So we've now all moved into the Institute for Neural Dynamics. And I want to tell you just a little bit, just to give you a taste of what's happening, because the Institute has a bunch of different teams that are focused on a lot of things, but we still have a lot of this type of high-density electrophysiology, neuropixel probes, a lot of calcium imaging and optical physiology, not just calcium imaging, but using like fiber photometry to image different types of indicators and transmitters deeper in the brain. We're doing some calcium imaging that gets combined with spatial transcriptomics so we can get finer resolution on cell details, as well as some molecular anatomy teams that are working to kind of map some of the brain-wide circuits with much more resolution. So for instance, we use what's called an expansion-assisted light sheet microscope, where you can sparsely label cells and then clear and expand the brain so we can image the brain intact, and we can get full brain reconstructions of individual neurons, right? And so this gives us really high detailed information about the anatomy of individual cells and allows us to start thinking about the circuits. A lot of the connectivity work, like when I showed you that subway map of the different brain regions, is done on bulk, right? We put a bunch of indicator into one brain region, we look to where that goes. But you don't necessarily get a view of how individual cells, does every cell in region A all project to region B in the same way, or do we have a subset that goes to region B and a subset that goes to region C? And this type of morphology allows us to pull apart more discrete circuitry of the brain. And so we're collecting a lot of this type of morphology data to give us these single cell brain-wide reconstructions. You can see there's a lot of cells project to a lot of great regions. And we can use this to now kind of map out regions that are connected into specific circuits that we can now target for either electrophysiology or optical physiology, where we're no longer restricted to the cortex, we're no longer focused on the visual system, but really looking brain-wide. And we can combine this with optogenetics, we can combine it with a bunch of kind of behavioral paradigms that are focused a bit more on decision-making. And so kind of working on foraging tasks where mice are being asked to choose kind of where to forage for rewards. So these experiments are kind of just right now kind of up and running, and we're starting to collect a large amount of data, both in terms of the morphology, but also with this brain-wide anatomy. And we're taking a slightly different approach to how we share this data. So rather than kind of packaging everything up into a single data set with a bow on it and a set of software tools, our data is open kind of as we collect it and in our bucket. And as I said, we're kind of just getting started. So you're welcome to go, see what's in the bucket, but you're probably not going to be able to make heads or tails of it until things are kind of a little further along. But I just kind of wanted to put that on your radar so you could get excited about more of these types of observatory style data sets that are coming out in the future. All right, so I will wrap up there. I just saw a question over here, if you saw it.
Single cell morphologies.
Yeah.
Did you say you could sparse label neurons and then do expansion microscopy? Was that what you were showing?
Yeah, so it's sparsely labeled neurons, and again, this comes down to a lot of molecular biology, where there's a couple of different approaches that go on. Part of it is, we've got these promoters, and you can kind of do intersectional promotion strategies, so it kind of reduces, I can have a promoter for all of the layer 4 cells in cortex, but I can cross that with another promoter that's more selective, so it will reduce it. There's also tricks that, I have a degree in molecular biology, but I'm not a molecular biologist, so don't tell my college that. But there's techniques that basically just kind of make the expression more sporadic. So it for a given promoter, it will reduce it to maybe less than 10% of the cells actually express it. And so we use strategies like that to just, instead of expressing it in thousands of neurons in the brain, we're expressing it in 100. I might be off by a factor, but enough, it's reduced enough that you can actually trace individual neurons in the cortex. But,
do you trace manually?
Yeah, so the annotation is done manually, and so there's a lot of steps of stitching the images and registering them. We register it into a common coordinate framework so that we can actually say which brain regions all of the structures are in, and then we have manual annotation, and this is actually probably starting in a few months will be something that if you're I really care about brain structure X, we're going to have a tool where you can you can get on your computer and pick cells in that brain region that haven't been annotated and contribute that annotation into the data set so that we can, if you're waiting for us to do the annotation and we don't care about your brain regions as much as you do, you might be waiting a while. Whereas if you're I'm really motivated to do this annotation, we'd love to have you contribute and collaborate in that way. So we're developing tools to make that a possibility. All right, other questions? Yeah.
Is there any future plan to move to another species? Or other species, monkeys?
So we don't have plans to work in other species. We're really focused on the mouse. And the Institute for Brain Science doing the spatial transcriptomics, they're doing some cross-species comparison work. But on the physiology and behavior side, we just work in mouse. Yeah. Yeah.
Saskia, just a shout out. This is the most, I would say the Allen Institute and their efforts are one of the most remarkable achievements in neuroscience and stuff like that. So I mean, really great job. It really is incredibly impressive. If you haven't seen any like this, like the spatial transcriptomics, cell type library is just, it's mind blowing really. And also shout out, they put you had some data from Josh Siegel up there. Josh, I showed some of this data. And one of the points I was making in my, sort of my talk was sort of the imperative for embracing the kind of ethology, allowing microbes to do what they do, and keep discovering place cells by basically letting, taking the rodents out of the, sort of the head mount, allowing them to move freely. So thinking about the importance of naturalistic behavior, just wondering how you're thinking about incorporating that. In our own experiments, I showed some of our really visual cortical data where you see like spatial place-like fields and animals that can actually move and navigate. Our own experience, way back doing visual mapping, when we took what I refer to as like primate-centered view, displaying front, sort of forward-facing cues, distal cues. Similarly, got very kind of poor responses, but as soon as we put the cues down on the floor, that is the lower visual field, where a lot of the sort of rodent visual action is, dramatic change in visual responsiveness. That's where you see the spatial specificity. So I'm just sort of wondering how you're thinking about embracing, ethology, and really naturalistic behavior. You pointed to like foraging, and that's kind of great. Any other thoughts about how you might add that kind of behavioral complexity to understand the real function beyond the standard model, which is sort of a coding model? How do you represent objects? But in fact, the real model is how do I use visual information to guide behavior? Yeah,
no, that's a great question. And I think the things that we have going on are, so that these foraging behaviors are much more flexible, right? Like you can ask questions about foraging in a lot of different. The types of cues that are used to provide information can kind of span different modalities. And so one of the things that's going on there is that we've got like a couple of variants on these tasks that can use different types of stimuli as information. Some of them really kind of don't have any stimuli as information. Some of them are more virtual reality based, where it's kind of simulating a mouse exploring an environment where there's different cues in different spaces, rather than just kind of you've got left and right choices. And there are kind of preliminary plans to move into open field types of experiments, which I think will happen in the behavior before we get the physiology in there. But I think we're hoping, like our plan is to get the physiology, doing some open field physiology as well. So we definitely are moving in that direction, but it's a direction like, we're well over time. So, I have lots of thoughts and feelings about ethological behavior, and it needs to start on the behavior side, right? You really need to start these experiments behaviorally before then trying to go in and do the physiology and trying to understand kind of what all these different things mean. And that's kind of how we're doing it now, in large part because the technology is just more complex. But but that is a really important part of all of this, because all of these behaviors are really kind of are pretty arbitrary for the mice. So yeah.
Just a quick question. So is it a standard practice to do functional recordings of any kind of electrophysiology, plastic, whatever, before you do structural studies on the same mice?
Yeah. So we do have a couple of data sets where we've done calcium imaging followed by EM. And so one is part of what's called the Microns Consortium, where actually we didn't do the calcium imaging. A team at Baylor did the calcium imaging, and then we did the EM. And then Sebastian Song's group at Princeton did a lot of the image processing kind of part of it. And we have a second data set that's the same thing, but all done in-house, where we did calcium imaging with very similar stimuli as what we used here, not the full set, but a subset. And we imaged a cubic millimeter of V1. And so that takes like 30 days of imaging to get all of that, and then did EM on that tissue. And these data sets are monumental to collect, and then like the effort in collecting it is heroic. The effort to like process the data and like register is like like conceptually it's easier, but it takes so much longer. And so those data were recorded, I think, in like 2018, like the calcium imaging was done in 2018. We just have like maybe 50 cells co-registered between the EM and the calcium imaging. I might be underestimating that. It's maybe 200, right? But like there's thousands of cells in that volume. So there's been like a lot of analysis of the calcium imaging by itself, but that's not interesting compared to combining it with that structural data. So that data exists. I don't know that it's been released in any format, but I think just because we're kind of waiting for the EM to be to be able to co-register it better. But all that exists, and the microns data set is, if you haven't heard of it, it's a data set, like if you go Google search microns consortium, you'll find it, and that's been out and widely used. So.